This is the third part of our lecture series on chemistry review. We'll be talking about proteins today. So recall from our previous lecture, we've already discussed two of the four biomacromolecules, namely carbohydrates as well as nucleic acids. Proteins, they are also polymers and large biomacromolecules, and they composes about 10 to 30% of the cell mass, and it's the basic structural material of our body. However, not all proteins are structural materials. Many play other types of important roles in allowing the cells to function. So proteins are polymers made up of the monomers called amino acids, and there are about 20 types of amino acids. And then in our next lecture, or next part of the lecture, we'll then be discussing lipids. The building blocks of proteins are these monomers called amino acids, and there are 20 different types of amino acids. As shown in the typical amino acid here, all amino acids have two important functional groups. They all have a basic group called an amine group, which consists of a nitrogen bonded to two hydrogens. All amino acids also contain an organic acid group, specifically called a carboxyl group. Okay, let me type it in here. Carboxyl group, which is essentially a carbonyl group bonded and also with a hydroxyl group. So a carbonyl group is basically carbon double bonded to an oxygen. A hydroxyl group is basically this OH, okay? And together forming this carboxyl group, okay? An amino acid can act as either a base, in other words, a proton acceptor, okay, on this end, or it can act as a proton donor, in other words, an acid, okay? What you don't see illustrated here is that this nitrogen actually has two lone pairs of electrons, okay, that can accept another proton. So another hydrogen can be attached here, okay? In that case, it would make this serve as a base, accepting that proton. But this side of the amino acid can actually donate give away its proton here in the hydroxyl group. Okay, if it were to do that, this amino acid would now become negatively charged. Okay, and so in such a way, every amino acid essentially can function either as a base or as an acid. Okay, all amino acids are identical except for your R group. Okay, and it's this difference in R group that makes each amino acid chemically unique from one another. All amino acids, however, you can see they all contain carbon, oxygen, hydrogen, and nitrogen. Okay. The proteins have a very wide range of functions in our body. Some have a structural role, such as collagen. Okay, and the collagen is a protein that can be found in the hair, and fingernails, feathers, horns, cartilage, tendons, other connective tissues in our body. And there are also proteins that serve for immune protection to help fight off intruding pathogens. Okay. Others help with the coagulation of blood, so serving as clotting factors. Not written here, but they're clotting factors. Proteins are clotting factors that help with blood coagulation. We also have regulatory proteins that help control cell activity, and some of those include hormones such as insulin. And yet other types of proteins that we find in our body, some of them are contractile proteins that allow for muscle contraction to occur for the heart or the skeletal muscle to contract. And some of them allow the sperms to swim or allow the cilia on the lining of your airway to beat. Okay. And yet other proteins serve a transportation role. They help carry oxygen molecules around your body. Okay. And that's hemoglobin. So take a moment to pause this video here and make sure you can draw out uh, this diagram, this block diagram of amino acid in this form, but also in the um, organic chemistry shorthand. Okay. And we can do practice of that during class as well. This is the diagram showing the 20 naturally occurring amino acids that can be found. And notice that they all share that carboxyl group we talked about, as well as the amine group. Okay, so the basic end and the acidic end. And then they have that center carbon bonded to an R group, which is the variable part that differs across different amino acids. The simplest of all amino acids being glycine. The R group is really only just the hydrogen. Okay. And definitely take a look at the names of these amino acids, and you'll be able to recognize the names, these terms as amino acids. Alanine, arginine, asparagine, aspartic acid, sometimes also called aspartate. There's cysteine. Okay, cysteine is actually an amino acid that contains sulfur. Okay, and we also have glutamic acid, also known as glutamate. There's glutamine, there's glycine, histidine, isoleucine, there's leucine, lysine, methionine, phenylalanine, proline, serine, threonine, tryptophan, tyrosine, and valine. Okay. So the two sulfur containing amino acids are cysteine and methionine. Just another way of depicting amino acids. So make sure that whether it's drawn in organic shorthand form or in this form, you should be able to recognize this as an amino acid structure. And practice drawing out amino acid structures. This is a list of abbreviations for the 20 amino acids. Okay, so take a moment to pause this video and go over these to make sure you know the abbreviation for each of the amino acids. Proteins can be described in terms of 4 structural levels: primary, secondary, tertiary, and quaternary. The linear sequence of amino acids in the polypeptide chain is called a primary structure of the protein. And it can be compared to the sequence of letters that spells a specific word. So primary structure is just the sequence of amino acid. Okay. It may be proline, valine, aspartate, so that specific sequence. of those amino acids lined up in that specific sequence. Proteins don't normally exist as just simple linear chains of amino acids. Instead, they twist or bend upon themselves to form more complex secondary structures. The chain begins to fold as the side chains come together and the hydrogen bonds form between various atoms within the chain. And the two most common patterns of hydrogen bonding between amino acids cause the chains to either twist into a corkscrew-like shape called an alpha helix, or form these zigzag folding sheets called beta pleated sheets. And so this hydrogen bonding between amino acids gives a protein its secondary structure. Now, a single polypeptide chain, okay, which is a really large molecule, really long sequence of amino acids, and it's considered a single molecule. And now within the single molecule, we can find both types of secondary structures at various places along its length. Many proteins have tertiary structure, which is the next higher level of complexity, and this can be superimposed on the secondary structure and involves the amino acids R groups. Okay, and so tertiary structure is achieved when an alpha helical or beta pleated regions of that polypeptide chain fold upon one another to produce a compact ball-like or globular molecule. Okay, and the side chains or the R groups interact in such a way that hydrophobic R groups, there are certain R groups in some of the amino acids that will tend to want to hide themselves inside the molecule. So the hydrophobic R groups are going to try to go on the inside of the molecule, whereas the hydrophilic R groups are going to try to be exposed on the outside. And so the tendency for those different R groups is a main driving factor that determines the tertiary structure. And so their interactions plus those reinforced by the covalent and hydrogen bonds will help to maintain that unique tertiary shape. Now, some protein molecules also have a quaternary structure in which two or more polypeptide chains are held together by bonds between the amino acids on the different chains. So it's important to note that in a quaternary structure, this now involves different molecules, okay? Shown in different colors here. This is a polypeptide chain. This is another polypeptide chain. This is another polypeptide chain. And these peptide chains are separate molecules, and they can now be assembled into a larger protein through these other weaker interactions. And this is an example, our example of this is hemoglobin. Hemoglobin, for example, is made up of 2 alpha chains and 2 beta chains, okay? And so a quaternary structure is essentially when 2 or more polypeptide chains are held together by bonds between the amino acids on the different chains. So think about what determines how a protein structure is folded. Even though these structures look kind of chaotic and make you think that it's sort of just random and by chance. So although a protein with the tertiary or quaternary structure looks like a clump of angiopasta, the ultimate overall structure of the protein is actually very specific, and it's very much dictated by its primary structure. In other words, the types and relative positions and exact sequence of amino acids in the protein backbone determines where those weaker bonds like hydrogen bonds and van der Waal forces, which you may have learned about in chemistry, can form to produce the complex coiled or folded structure that keep the water-loving amino acids near the surface and the water-fearing amino acids buried in the protein's core. Okay. A slight change in subject. We'll be describing most types of body proteins in conjunction with the organ system or functional processes to which they are closely related. However, there's one group of proteins, enzymes, that's intimately involved in the normal functioning of all cells. So we'll be considering these incredibly complex molecules here. Enzymes are globular proteins that act as biological catalysts. Catalysts are substances that regulate and accelerate the rate of biochemical reactions, but they themselves are not used up or changed in those reactions. You can think of enzymes as chemical traffic cops that keep our metabolic pathways flowing. Enzymes cannot force chemical reactions to occur between molecules that would not otherwise react. So the three basic steps that are involved in enzyme action. First, substrate or substrates will bind to the enzyme's active site, and that temporarily forming an enzyme substrate complex. Substrate binding causes the active site to change shape so that the substrate and the active site fit together precisely and in an orientation that favors reaction. In the second step, the enzyme substrate complex undergoes internal rearrangements that form the products. The enzyme then releases the product or products of the reaction and then returns to its original shape. And again, available to catalyze another reaction. And a few things to note about enzymes. Enzymes are very choosy. They bind only with their appropriate substrate molecule, much like a lock that can be opened with only one key. And second, the exposed atoms in an active site have electrical charges that attract rather than repel the substrate molecule. And only the substrate molecules can fit into the active site's groove. Okay. And once the substrate is bound to the active site, a reaction can take place, and that usually happens very quickly. Although enzymes are specific for particular substrates, other non-substrate molecules may act as enzyme inhibitors if their structure is similar enough to occupy or to block the enzyme's active sites. And because enzymes are unchanged by their catalytic role and can act again and again, cells typically only need a small amount of each enzyme. And that catalytic reaction occurs with incredible speed. Most enzymes can catalyze millions of reactions per minute. Misspelled proteins can occur when the sequence of amino acids is incorrect. If the enzyme is altered even the slightest bit, that may lead to a change in active site, and the enzyme may no longer be functional. Slightly modified non-functional proteins or slightly modified non-functional enzymes are responsible for a large number of diseases and physiological problems. As you already know, chemical reactions have reactants and products. The products are formed from the reactants. And for reversible reactions, the products can also be seen as the reactant when viewed in the opposite direction. Okay. And many reactions will occur in both directions, and they proceed predominantly in one direction or the other based on reactant and product concentration. Enzymes speed up the rate of chemical reactions to make them compatible with life. we will not be able to eliminate the waste carbon dioxide produced in our tissues without carbonic anhydrase. So carbonic anhydrase is an enzyme that dramatically speeds up the conversion or interconversion between carbon dioxide and water to carbonic acid and in reverse. Okay. Now we're moving on to talking about protein synthesis. As we've learned, proteins are made up of monomers called amino acids, and this is the general structure of amino acids. Recall that we've got 20 different types of amino acids, and but all of these amino acids share similar structure. The way they differ is in this R group. All amino acids have a carboxyl group, okay, and also amine group. Okay. And we're gonna take a moment to watch a short video on how protein synthesis occurs.
Here is a cell, the basic unit of all living tissue. In most human cells, there is a structure called the nucleus. The nucleus contains the genome. In humans, the genome is split between 23 pairs of chromosomes. Each chromosome contains a long strand of DNA, tightly packaged around proteins called histones. Within the DNA are sections called genes. These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of free bases in the nucleus. The DNA code determines the order in which the free bases are added to the messenger RNA. This process is called transcription. Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA. The messenger RNA then moves out of the nucleus into the cytoplasm. Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA. The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids. There are 20 different types of amino acid. Transfer RNA molecules carry the amino acids to the ribosome. The messenger RNA is read 3 bases at a time. As each triplet is read, a transfer RNA delivers the corresponding amino acid. This is added to a growing chain of amino acids. Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein.
So peptide bonds between amino acids are formed in dehydration synthesis reactions. We've discussed dehydration synthesis in hydrolysis before, and if you recall, in dehydration synthesis, the acid group of the one amino acid is bonded to the amine group of the next amino acid. And in this process, there's a loss of a water molecule. And this process requires energy in the form of ATP. And so, again, you see that to form a bond between these 2 amino acids, to link these 2 amino acids together, you get the hydroxyl group of this amino acid being linked to the amine group of the next. And this forms a bond we call the peptide bond. Okay, so essentially a peptide bond is formed between where the hydroxyl group used to be and between that carbon and the nitrogen of the amine group. The reverse reaction, a hydrolytic reaction, occurs when we break a dipeptide into two singular amino acids. And in that reverse reaction, the peptide bond that links the two amino acids together is broken, and water is required for this reaction to occur. The proteins are made by ribosomes. Ribosomes will link amino acids together in the order that's specified by the mRNA or messenger RNA molecule, as we shall see very soon. And so in this image, you can see that the ribosome essentially reads off the code on the mRNA sequence and use it as a template. And it then pulls in the tRNA, corresponding tRNA. Each tRNA is linked to an amino acid. And as the tRNA comes in, it allows the addition of yet another amino acids to this growing polypeptide chain. The central dogma of molecular biology describes the two-step process by which the information in genes flows into the protein, namely from DNA to RNA to proteins. And this process of building a protein from DNA sequence is not a direct one. Okay, you can see that it incorporates the middleman RNA, which is also a nucleic acid. So polypeptide synthesis essentially involves two major steps: converting from DNA, going from DNA to RNA. Okay, this process is called transcription. And this is where the DNA information is encoded to mRNA. And then the second step, where the information carried by mRNA is now decoded and used to assemble the polypeptide. This process is called translation. Okay, so we've got transcription followed by translation. This is a video we've already watched. I've just included it here again for those of you who want to review it. This is just a repeat slide. So again, going from DNA to RNA, this process is called transcription, and RNA to protein, this process is called translation. And we can see that in the process of translation, as we've already discussed, this process involves building this polypeptide chain by decoding the sequence on the mRNA. Now, let's take a look at where transcription and translation occur. You can see here that transcription, encoding that DNA sequence into mRNA from the RNA, this process occurs inside the nucleus. And then after the mRNA is made, it undergoes some processing and modifications, and then it's exported out of the nucleus into the cytoplasm. And in the cytoplasm, its sequence is then read by the ribosome, which then decodes that sequence and creates that polypeptide chain, which later folds into a protein. Okay, so translation, in other words, occurs inside the cytoplasm. Okay, and this is key because if we are to have to pull the DNA out of nucleus every time we need to make a protein, okay, it would make the DNA a lot more susceptible to damage, mutation, and that's not good because we want that that genetic code to stay in a place that makes it less susceptible to damage. We want to be able to create the same proteins in the correct sequence again and again and again, right? And cell divisions after cell divisions. So we want to make sure it's in a sacred place where it's a well protected. And so in using this middleman mRNA, we're able to carry that message out into the cytoplasm without damaging the DNA. So the general gist of this transcription and translation is that inside the nucleus, the RNA polymerase will bind to the template strand of DNA and then transcribe the DNA sequence into mRNA. mRNA undergoes modification before it exits out into the cytoplasm. And in the cytoplasm, ribosome then translate that mRNA sequence into a sequence of amino acids that eventually form the protein. Now, there's a lot of stuff happening in this diagram. We're going to look at the details now. Okay, so once again, RNA polymerase will bind to the template strand of DNA, and then as it reads that template strand, it transcribes that sequence into RNA transcript. As you can see, this RNA transcript is formed from the 5' end to the 3' end. Okay. After this mRNA or pre-mRNA is made, it's then spliced and modified to produce the final mRNA. Okay, you can see that there are segments called exons and the segments called introns. Essentially, the introns are cut out, okay, and the exons are spliced together, and they are what eventually exit out into the cytoplasm. Besides that, okay, there's also a few other modifications that happens. Addition of a poly A tail, okay, and also addition of a 5' cap, okay. And so through these modifications, the mRNA is now ready for transport out of the nucleus. After it enters into the nucleus, after it exits out of the nucleus, okay, it now attaches to ribosome. The ribosome subunits will assemble around the mRNA transcript. Meanwhile, we have amino acid activation where each amino acid attaches to its proper tRNA with the help of specific enzyme as well as ATP. So this process requires energy. Okay, because we're linking things together. Okay, and this process is facilitated by an enzyme called the aminoacyl tRNA synthase. Okay, in either case, in the next step, okay, after ribosome has assembled around the mRNA transcript, and we got these amino acids primed to the tRNA, we can now start the process of translation. And a succession of tRNAs will add their amino acids to the polypeptide chain as the mRNA is moved through the ribosome, one codon at a time. And when this is completed, the final polypeptide is then released from the ribosome. And after that, this long chain of amino acids can now begin folding and into the globular structure that becomes the functional protein. So you can see here, there are 3 forms of RNA typically that work together to carry out DNA's instruction for polypeptide synthesis. That includes messenger RNA or mRNA, which are single-stranded nucleotide chain that's made based on the DNA template strand. and it carries the coded information to the cytoplasm where protein synthesis occurs. We also have ribosomal RNA or rRNA. Along with proteins, these rRNA form the ribosome. Okay, and the ribosome consists of two subunits. There's one large subunit and one small subunit that combine to form the functional ribosome. Okay, and ribosomes the site of protein synthesis. Finally, we also have tRNA or transfer RNA. These are small clover-shaped molecules that carry amino acids to ribosomes. That's where they decode the mRNA messages or amino acid sequence in the polypeptide to be built. We're now going to take a look at this short video.
This student is cramming for her biology test with only a frosting-covered donut for lunch. Specialized cells in her pancreas respond to the increasing amount of sugar in her blood by releasing insulin, a small protein that regulates blood sugar levels. Let's go inside one of these cells to see how this protein is manufactured. The instructions for making insulin are coded by a segment of DNA in the nucleus. In transcription, an enzyme zips along the DNA, forming RNA, shown here in red. RNA nucleotides line up with their complementary DNA partners, transcribing the information in DNA into RNA. As the RNA grows, it is processed in several ways. First, a modified guanine nucleotide is added to the beginning as a cap. Also, segments of the RNA strand that do not actually code for the protein are removed, and the remaining segments of RNA are reconnected. Finally, extra adenine nucleotides are added to the end of the RNA strand, forming a tail. The completed messenger RNA, mRNA, now leaves the nucleus. The message in mRNA is translated into a protein in the cytoplasm. First, a transfer RNA, tRNA, arrives, carrying a specific amino acid. The small subunit of a ribosome attaches to the mRNA. Now, the large subunit of the ribosome attaches. A second tRNA docks, bringing another amino acid. The ribosome helps to form a covalent bond between the two amino acids. The mRNA shifts, and the first tRNA leaves. A new tRNA brings another amino acid. The ribosome helps to form a new bond, and the process is repeated. Notice that one end of a tRNA molecule has a set of 3 bases called an anticodon that pairs with complementary bases on the mRNA. The other end of the tRNA carries a specific amino acid. Different types of tRNAs carry different amino acids. In this way, the message in mRNA is translated into a specific sequence of amino acids. For proteins that will be secreted from the cell, like insulin, the ribosome docks on the rough ER, and the protein grows into the ER compartment. The new protein molecules are packaged in a vesicle that is transported to the Golgi apparatus, where many proteins are processed. However, insulin is packaged in a vesicle that leaves the Golgi and is then processed. Proteins secreted from the cell are shipped to the plasma membrane. Here, insulin is secreted from the pancreas and begins to regulate the student's rising blood sugar levels.
We're now going to go over this process from transcription to translation in details once again. So the first step Okay, protein synthesis is that transcription. Okay, the nucleus contains our DNA, and each chromosome can be seen as one molecule of DNA. And within each molecule of DNA, there are genes. And for the purpose of our discussion, we define genes as a segment of DNA molecule that carries instructions for creating one polypeptide chain. There are some cases where some genes actually specify the structure of certain varieties of RNA as their final product, but just for the purpose of our discussion, we're gonna see gene as a segment of DNA that carries information for creating one polypeptide chain. So RNA polymerase is the enzyme that is most important in the process of transcription. It will attach itself to a specific sequence on the template strand of DNA. It then move along that DNA template strand and building mRNA from free nucleotides based on the sequence of DNA. So the DNA sequence essentially determines the order of bases that are linked together in the mRNA. Okay. The pre-mRNA that's being transcribed is now ready for processing. So it undergoes modifications and splicing before the mature mRNA is formed. The mature mRNA then moves out of the nucleus into the cytoplasm, where it's then attached to ribosomes. In the cytoplasm, the ribosome binds to the mRNA transcript and then begin reading it 3 bases at a time. And that triplet of mRNA bases is called a codon. And we also have anticodon. That's basically a triplet of corresponding 3 bases on the tRNA. Okay, so the anticodon on the tRNA corresponds with codon on the mRNA. And with the tRNA coming in, it then brings along with it an amino acid, and this amino acid then is linked to the growing polypeptide chain. So transfer RNAs or tRNAs, they are the type of RNA that's responsible for carrying amino acids to the ribosome. And you can see that it has a clover leaf shape to it. Okay. And on it, we have the anticodon, okay, which is a sequence of 3 bases. Okay. So this RNA codon table will allow us to determine the sequence of amino acids based on the mRNA sequence. And what you are responsible for on this slide is you should know how to read this table, and you should also know how to identify the first amino acids on the mRNA sequence, as well as on the template DNA strand and coding DNA strand. So how to read this table? Well, for example, you come across the codon ACG, A, C, G, we know it encodes for the amino acid glycerol. Okay. And I also said you should know how to identify the first amino acid on the mRNA. The first amino acid on the mRNA, well, you're gonna see a string of letters, basically alternating between these 4 letters, A, C, G, and U. Okay, and so when you come across a sequence A, U, G, okay, you know that's going to be the starting point. And also note that this is going to be read in the 5' to 3' direction. The mRNA sequence is read from the 5' to the 3' direction. Okay, and so when you read from the 5' to 3' direction and you see the sequence AUG, you know that's going to be the starting point. Okay, and so this sequence is going to encode for the amino acid called methionine. And so AUG also is called the start codon. Okay, there are 3 stop codons. Okay, UAA, UAG, UGA. Okay, and we'll do practice for this This is just another form of the RNA codon table where you start the center of the wheel and then work toward the outside.